Bismillahirrahmanirrahim. Assalamu alaikum dear students, welcome to your channel. In today's Embryology Foundation module lecture 11, we are going to complete the development in the third week of development, and the learning objective for the today's class are the establishment of the body axis, to understand the clinical correlates of the gastrulation, and to understand further development of the trophoblast. So what about establishment of the body axis is that establishment of body axis, that is anteroposterior or craniocaudal, left, right, and even the dorsoventral are even established at early blastocyst stage. Main factor involved is the anterior visceral endoderm. So anterior visceral endoderm is present at the cranial end of the bilaminar germ disc, and this endoderm is activated by the factors the OTX2, LIM1, HESX1, and secreted factors the cerebrus and lefty1. So these all factors maintain the anterior visceral endoderm, and this anterior visceral endoderm is responsible for the formation of the head region. In the absence of the factors, the lefty one and the cerebrus, the primitive streak is maintained, and this primitive streak will continue to grow and will form all the three germ layers will be continued to form over here. So three factors involved for the anterior visceral mesoderm are these are the transcription factors, the OTX2, LIM1, and HESX1, and the secreted factors are the cerebrus and lefty one. In addition to this, what happens to the mesoderm, that the fibroblast growth factor and the bone morphogenetic protein, these both the factor bone morphogenetic protein is basically secreted throughout the embryonic disc, and this bone morphogenetic protein along with the fibroblast growth factor will ventralize the mesoderm. So in this diagram you can see this is a trilaminar germ disc. In this you can see the neuroectoderm is present, which is going to form the neuroectoderm. This area is the notochord, then the paraxial, intermediate, and the lateral plate mesoderm, and then the endoderm is over here. So what is the meaning of the ventralization of the mesoderm is that the cells which are going to form mesoderm under effect of FGF and BMP4, they are ventralized, means they are going to form intermediate mesoderm to form the kidneys and also the lateral body wall structures. So in fact all of the mesoderm will be ventralized if there is no check and balance for FGF and BMP4. But actually this doesn't happen. There is a check and balance at the level of the node, and this node secretes factors which are the caudin, nogin, and follistatin, and these factors are activated by the factor glucosid. So these factors antagonize the activity of the BMP4, and because of this antagonization, then the mesoderm will be dorsalized. So this dorsalized mesoderm will form later on the notochord, the somites, and somatomeres. So in this way, under the effect of FGF and BMP4, mesoderm is ventralized, and by antagonizing the activity of BMP4, it will be dorsalized to form notochord and somites and somatomeres, and later on will have an inductive effect on the ectoderm for the process of the neurulation. So these factors are very important, and main organizer at this site is the node. So this node secretes different factors, and it organizes the expression of the different proteins. Another important factor is the HNF3 beta protein, which maintain the nodal factor which is secreted and the primitive node. And this HNF3 beta is important because in absence of HNF3 beta protein, there is underexpression of the factor nodal, which will lead to underdevelopment of the forebrain and midbrain region in the embryo. So these are different factors which is related to either endoderm, that is anterior visceral endoderm, and then which are the factors OTX2, LIM1, and HESX, along with cerebrus and lefty one. In addition to this, for the mesoderm, there are different factors. So for ventralization, the FGF and BMP4 is involved, and to dorsalize the mesoderm, we have three factors: the caudin, nogin, and follistatin, which are activated by gossychide and will then antagonizes the activity of the BMP4. As a result of antagonization of the BMP4, then the mesoderm will be dorsalized to form the notochord, the somatomeres, and later on will induce the ectoderm to form neuroectoderm, and then neural tube development will occur. So what happens in the mid region and the caudal region? Then there is a role of the brachyury T gene over here, and this brachyury T gene is acting in combination with the TOTA genes, and they regulate the dorsal mesoderm formation. So this is also very important, and we will see a little bit later in the next slide that the effect in the brachyury T gene will lead to deficiencies in dorsal mesoderm formation and will ultimately form the caudal dysgenesis syndrome. In addition to all these factors, the laterality is also induced by FGF, which induces the expression of the node. So up till now we are studying the cranial and the caudal axis, in which anterior visceral mesoderm, anterior visceral endoderm was involved, and also the dorsalization and ventralization of the mesoderm. And now we are going to study the laterality, the right and left sidedness, how they will occur. So how the laterality is established? So you must remember that the factor nodal is over here, and this factor is secreted by the node, and this factor causes the accumulation of the 5-hydroxytryptamine over here. So 5-hydroxytryptamine, also called the serotonin, when it accumulates on the left side of the embryonic disc, it will activate a factor that is called MAD3 factor, and MAD3 factor will be responsible for the maintaining all of the 5-hydroxytryptamine to the left side. Similarly, this nodal factor also activates the PITX3 factor through the effect of the LEFT2 gene, and this PITX2 factor is master gene for the left-sidedness. So you must remember that there is a right and left asymmetry in the embryo. Some organs are present on left side, like the heart and spleen, and some organs are present on the right side, like the liver, appendix, etc. So this asymmetry is maintained by the the factors which are secreted at the nodal. So first, nodal will stimulate the release of the 5-hydroxytryptamine, and this 5-hydroxytryptamine will later on will causes the accumulation of the, activation of the MAD3 factor, and MAD3 factor will be responsible for all of the secreted factor to remain on the left side. Similarly, three other factors are also involved. One of these factors is the LEFT1 factor, another factor is sonic hedgehog protein, and the third factor is the zinc transcription factor, also called ZIC3. So not only these three factors maintain the midline structures of the embryonic disc, but they also restrict the factors on the left side, as a result of which left-sidedness is maintained. So remember, this is very important point. And another important point over here is at the nodal, at the level of the node, primitive node, there are the cilia which are present, and these cilia beat towards the left side, and they beat and they causes movement of the nodal and 5-hydroxytryptamine to the left side. This is also very important clinically, because we are going to discuss some clinical correlates. So we will discuss over there the activity of the cilia which are present over node. So again, to summarize, the nodal is secreted, which causes accumulation of 5-hydroxytryptamine. This 5-hydroxytryptamine will activate the MAD3. Similarly, nodal to activate the LEFT2 gene, and LEFT2 gene will activate the PITX2 gene. This PITX2 gene is a master gene for the left-sidedness. At the same time, the LEFT1 gene, the sonic hedgehog gene, and the zinc transcription factors which are released, these not only maintain the establishment of the midline structure, but also they restrict the movement of the nodal and 5-hydroxytryptamine to the left side. So please also remember, we are continuously talking about the factors which are present on the left side. So far we do not know the factors which are released on the right side and which are responsible for the right-sidedness of the embryo. So now we are going to discuss the clinical correlates with the process of the gastrulation and the problems with the establishment of the body axis. So first is the holoprosencephaly. Prosencephalon, as you all know, is the forebrain. So the reason behind holoprosencephaly is the high doses of the alcohol, because this is going to happen at the time when the patient just misses her first menstrual periods after the conception, and she even does not know that she is pregnant. So she is not taking any precautionary measures. She may take high doses of alcohol that can lead to the holoprosencephaly. So in this way, the germ disc is damaged in the anterior midline. This is normal, and this is the holoprosencephaly. In anterior midline there is a small forebrain and also fusion of the lateral ventricles, and you will see the single ventricle over here, and the eyes will be closer to each other, the condition called hypotelorism. So these are the different features which are present in holoprosencephaly, in which underdevelopment of the forebrain along with the fused lateral ventricle and hypotelorism because of high doses of the alcohol. The second is the caudal dysgenesis. We also called it as sirenomelia. It is because of insufficient mesoderm in caudal most regions of the embryo. So as I have told you in the previous slides, that the Brachyury gene is involved, and this is responsible for the mesoderm in the caudal and the mid region. So if insufficient mesoderm is present, that can lead to the caudal dysgenesis syndrome. In this caudal dysgenesis syndrome, there will be abnormalities in the lower limb, there will be abnormalities of the urogenital system, and also the abnormalities in the lumbosacral vertebrae. So next is the sacrococcygeal teratoma. Sacrococcygeal teratoma is usually formed because we know that the primitive streak after the process of gastrulation usually disappear at the end of the third week of development. But if it persists and it completely do not disappear, it will lead to the sacrococcygeal teratoma. So as primitive streak is capable of forming all the three germ layers, so the sacrococcygeal teratoma contains the derivatives of all the three germ layers. In addition to this reason, sacrococcygeal teratoma may also result if the primordial germ cells do not migrate to the region of the their primitive gonads and they remain in the midline. So they can also lead to the sacrococcygeal teratoma. Other clinical correlate which are usually related with the gastrulation as well as the left-right sidedness and establishment of the body axis are. So first you should understand what is situs solitus. Situs solitus, that means that all the viscera, they are present at their normal side. The viscera which are usually present on left side, they are present on left side. So this is normal development. In case of situs inversus, all the viscera are present in opposite direction. So the heart on the right side, the appendix on the left side, the spleen on the right side. So there is a complete reversal, and this is the mirror image of the situs solitus. Another condition is a situs ambiguous. So in situs ambiguous, you should understand that it is not a complete situs inversus, but there is a condition in which some viscera are present on opposite side. In this case, there may be the isomerism of atria in heart, or there may be inversion of the atria in the heart. And this situs ambiguous can lead to either left-sided bilaterality or right-sided bilaterality. In cases of the left-sided bilaterality, there will be polysplenia. The spleen will be present on both the sides. And in cases of the right-sided bilaterality, either hyposplenism or asplenia will be present. Another important clinical correlate is the Carter-Jenner syndrome. This is also called the abnormal cilia syndrome. So as I have told you in the previous slide, that at the level of the primitive node, there are the cilia which are present, which beat towards the left side and responsible for restriction of the nodal to be present on the left side. So if in Carter-Jenner syndrome, which is also called abnormal cilia syndrome, all the cilia which are present in the body are affected, including the cilia at the level of the primitive node. So Carter-Jenner syndrome can lead to situs inversus, and in addition to situs inversus, in Carter-Jenner syndrome there will be bronchiectasis, because at the level of the bronchi there are the cilia which are affected. And in also female patient, the uterine tubes epithelium is lined by the ciliated cells, and this cilia moves towards either towards the uterine cavity after fertilization or before fertilization towards the ovaries. And if these cilia are affected in females in Carter-Jenner syndrome, there will be either infertility or subfertility. So this Carter-Jenner syndrome is also important from your MCQ's point of view as well. And next is the role of the antidepressant. So as I have told you, that serotonin or 5-hydroxytryptamine is very important and accumulates at the level on the left side, and it maintains the left and right sidedness. So in case of the patient who are taking antidepressant, which are serotonin reuptake inhibitors, will lead to deficiency in serotonin and will affect the left and right side, and it can lead to either situs inversus or situs ambiguous. So now we move on to last topic of the today's slide, which is also occurring and happening at the end of the third week of development. So you can see these are the cytotrophoblast and the syncytiotrophoblast which are present over here, and these are the lacunar spaces, and in lacunar spaces the maternal blood is present. So this area section taken from here, the core is formed by the cytotrophoblast and it is covered by the syncytiotrophoblast. It is called the primary villus. Very important viva question. You must know primary villus consists of the core of the cytotrophoblast, which are covered by the syncytiotrophoblast. So what happens next, that during the process of development in third week, when extra embryonic mesoderm is forming over here, this extra mesoderm also enters into the core of the primary villus, and this villus is now called the secondary villus. And when this extra embryonic mesoderm differentiate into the blood vessels and blood cells by the process of angiogenesis, so the villus is now called tertiary villus. So when the blood vessels are formed in tertiary villus, at the same time these blood vessels are formed in the chorionic plate and they are connected to the blood vessels of the villus, and also through the connecting stalk these blood vessels are attached to the blood vessels of the embryo. So when heart starts to beat and it pushes the blood will enter through the fetal blood vessels through the connecting stalk into the vessels of the extraembryonic membrane into the chorionic villi, tertiary villi. So you should also understand another point, that at the same time when tertiary villus is forming, also cytotrophoblast migrate through the syncytiotrophoblast and they come to lie at the junction of this maternal endometrium, and they form a shell-like structure over here which is called the cytotrophoblast shell. Cytotrophoblastic shells help in embedding and implantation of the whole structure into the maternal endometrium. Also important another point, that the villi which are present at the level of this embryonic disc and chorionic plate, and they are present and they reaches up to the level of the cytotrophoblastic shell. These are called primary or anchoring villi, and the villi which are present which branch from these primary or anchoring villi, these are called the free or the floating villus. So dear students, here I complete my today's class, and at this class, with this class, the third week of development is also completed. So we will continue inshallah in the next class with the neural tube development, and the references for today's class are Langman Medical Embryology, The Developing Human by Keith L. Moore. Thank you very much for your attention. Till the next class. Allah Hafiz.